I'm Dr. Sam Charlton. I'm a postdoc in the Environmental Microfluidics and Bio2M group at ETH Zurich. And I'm here to present a poster today of some work that I've been up to over the past couple of years. It relates essentially to biofilm susceptibility testing. So we know that there's many different varieties of tests for planktonic assessment of antimicrobial efficacy. And there's also emerging biofilm susceptibility tests. However, one thing that the current tests lack is real-time dynamic information on the poroelastic and permeability properties of bacterial biofilms. So the reason why bacterial biofilms are distinct from planktonic cells is that they form an extracellular matrices in which they encapsulate themselves within. And this results in enhanced stability against dynamic shear forces in the local environment. And it also results in changes to cell physiology deep within the biofilm and the formulation of self-produced gradients or gradients produced through the environment. And what the platform that we've designed here aims to do is capture these different dynamic processes going on in the biofilm system, quantify them using computer vision and mechanical testing, and provide more resolved, enhanced data on antimicrobial susceptibility tests against any different type of bacterial biofilm you can imagine. And the way that we accomplish this is through a microfluidic technology, which we've been working on over the past couple of years. And this involves the formulation of porous membranes within a microfluidic channel. Then what we're able to do is seed these membrane with an inoculum of cells. The density is the investigator's own distinction. And then what we do is we track biofilm formation with time within this localized area. And the poster here today is giving you a very solid idea of the fundamental applications of the technology on model systems. So the first part of the poster, we measure the hydraulic resistance of different wild-type biofilms. So in this part of the study, we looked at Pseudomonas aeruginosa, E. coli, Bacillus subtilis, and Staphylococcus epidermidis. And we were able to show that generally with increasing pathogenicity, you get reduced permeability of the bacterial biofilm. And what this means is that essentially external solutes will diffuse slower through the bacterial biofilm, meaning that there's a possibility that there's increased self-produced gradients within the biofilm, which could have a negative antagonistic effect on antimicrobial treatment. So not only can we track the formation of the biofilm with time and its morphological development, we can also track its dispersion behavior at the end of its cycle, essentially. And we can do this using a multitude of different phase contrast modalities. And this enabled us to characterize the different types of dispersion dynamics which these species had. So this could be useful for investigators out there wanting to look into dispersal-inducing treatments, for example. Next on, we looked at the influence of the composition of the extracellular matrices on biofilm permeability and stability using a Bacillus subtilis system. And within this system, we tested 3 different mutants, a polysaccharide deletion mutant known as EPSO, an amyloid fibril protein known as TAS-A, and then a triple mutant, which was devoid of both of these components and a hydrophilic surface coating all on the outside of the biofilm. And we're able to show that within our system, we're able to detect these subtle changes within the composition of the matrix, further confirming the repeatability and precision of our methodology. After that, we then tested inoculum effect. So this was done on Staphylococcus epidermidis. And in here, we tested a rich and a slightly weaker media upon the formation of the permeability of Staphylococcus epidermidis biofilm. And what we were able to do here is show that generally with weaker media, you get stiffer, more stable, lower porosity biofilms, which is reasonably significant, as if you're doing a standard plate test, we know that there's inoculum effects, and one of these could be essentially the change of the permeability of the extracellular matrices. So we're able to quantify that reliably within our system. And furthermore, within this, we also developed a analysis methodology using something called optical flow, which is where we essentially measure the cross-correlation between two images in a time series. And from that, we're able to derive the vector field of movement within the biofilm as it grows. And using this analysis, we're able to localize growth regions within the biofilm substructure. So we know that biofilms are also incredibly heterogeneous in their phenotype. And this is essentially a method in which we're able to pick out separate parts within the bacterial biofilm, where essentially we still have cell activity. And within this, we then calculated what we call like the penetration length of our treatment within our biofilm. So not only are we able to quantify the amount of killing biocidal effect going on, we're also able to measure the penetration range within the biofilm at which the treatment is having an effect. And this is quite cool because then this enables us downstream to then model the gradients within the biofilm, superimpose our penetration lengths, and then data mine essentially what kind of gradients are causing the failure of the treatment. And last but not least, we looked at Pseudomonas aeruginosa. And in this study, we essentially just wanted to validate our algorithms looking on cell death killing and quantifying the effect of extracellular DNA on the extracellular matrices permeability. So for this, we used PA14, which is a model aeruginosa organism, and we treated it with some inhibitory concentrations of ciprofloxacin, an antibiotic known to cause the release of extracellular DNA within this biofilm. And using these experiments, we were able to show that upon extra release of extracellular DNA, you essentially get a permeability reduction within the biofilm. And we theorize this is essentially due to cross-linking between the DNA and cationic polymers within the extracellular matrices. But yeah, this is essentially a way of validating our quantification of extracellular DNA within the biofilm with time, and then correlating that with the mechanics of the system. So I hope I've not overwhelmed the viewers with too much information, but this is a kind of proof of concept for our device. And at the moment, we're essentially developing a 2.0 version of this. So the idea is to have a 16 channel system, so we can run 16 conditions in parallel at the same time. And then this is also to be integrated with pneumatic controllers, enabling us to have a fully automated system which has intelligent experimental design as well, enabling the user to then program in the type of susceptibility study that they want to do, whether this is cycling through different treatments, whether this is cycling through shear rates. We aim to offer a platform which can produce this. So you can reach me on my institutional email, which you'll be able to find on Google. So yeah, any questions or any ideas, get in contact with me.